Thank you so much for joining this afternoon's session. Today we'll be discussing technology for monitoring and evaluation. I'm excited for the array of presentations that we have and the representation from UNC. So I'd like to introduce Ethan Chou, who is a student at the University of North Carolina and will be describing to us today his progress on improving semi-quantitative screening of total lead concentrations in drinking water using affordable test strips. Come on, meet them.
Yeah, as she said, I'm Ethan. I'm a senior at the University of North Carolina at Chapel Hill. I'm studying environmental health science, and here's my project on improving the screening of lead concentrations in drinking water using affordable test strips under the tutelage of Professors Northcross and Fisher. So, as many of you probably know, lead exposure is a serious health issue which affects many people all over the world. And one of the most common sources of lead exposure is through drinking water, which has been exposed to lead plumbing, which includes pipes from wells and municipal supplies most prominently, but also faucets and household plumbing. So these exposures have been linked to health issues through a large quantity of evidence, including stunted growth, IQ loss, and anemia, primarily in children, but also kidney damage, reproductive issues, and cancers later in life. So many methods of testing for lead in water, such as spectrometry, require prohibitively expensive equipment or specialized knowledge. At-home lead test strips are strips of paper which develop colored stripes when exposed to lead and are an affordable method to test for lead at home, as each strip averages around $15. So the variety we chose will develop two colored stripes on them in exposure to water. If the right stripe is darker than the left, then it has detected lead above the detection limit. However, there are two major issues which affect their ability to accurately report and measure lead in water. The first is that while they're able to report the concentration of dissolved lead, they cannot detect undissolved particulate lead, which limits the types of lead they can detect when particulate lead can also be found in water sources and also provides a health risk. So the second issue is that they only give a binary yes or no answer as to whether there is lead above the detection limit, and they don't give an actual number. So this makes it difficult to read the test strip results when the lead concentration especially is close to the detection limit, and adds like visual bias, which also deprives the user of information that could be used to determine how much risk they are in exactly. So a previous 2021 study by Chris et al. Found that adding certain household acids such as lemon juice and vinegar was able to dissolve particulate lead and improve their detection using these test strips. So based on this, we had two primary research goals. The first was to improve on this previous study to create a standardized method for acidification of water samples. And the second was to find a way to use digital image analysis to quantify these concentration readings. Okay, so first of all, we created water samples in sets of 5, each with a different lead concentration, 0, 1, 3, 10, and 30 micrograms per liter. The type of lead also varied between samples. We had dissolved lead using lead 2 chloride, small particulate lead using lead carbonate basic, and large particulate lead using shavings manually taken from a brass pipe. So the target concentrations were based on the estimated lead content of each substance. So for instance, we estimated the pipe shavings were about 3% lead by mass. So to get 30 micrograms per liter lead, we would use 1,000 micrograms per liter of brass shavings. So finally, we altered the acidification method between trials. We used unacidified samples to prove the baseline accuracy of the lead test strips. And then we used a method to acidify the water to 0.01 m citric acid and sodium citrate. And then each possible combination of these factors was measured 3 times. We primarily chose citric acid to reflect the lemon juice used in the Chris et al. Study, but also because it's a relatively safe acid that can be used in the household, even if it's not as strong as some of the other options. And sodium citrate was also added as a chelating agent to further improve detection. So first, to find the necessary incubation time to ensure that the acid could break down the particulate lead types, of half-liter solutions of lead carbonate or brass shavings were set on stir plates and acid was added. So at certain time intervals, a filtered sample was taken from each of them to remove the undissolved particulate lead, and So every 2.5 minutes for lead carbonate and every 10 minutes for brass. So 2 trials were done of each, and as can be seen in the graphs here, the proportion of lead dissolved by the acid reached its peak around 10 minutes for lead carbonate and around 20 minutes for brass. But we decided to proceed with an overall 40-minute incubation time for testing, both to just ensure that as much lead could be recovered as possible, and also because the maximum value for a brass occurred at the 40-minute mark. So we also took unfiltered samples of each before acidification, and then later acidified them with a large volume of nitric acid to provide a total lead concentration in the samples, which was verified with ICP-MS spectrometry. So the dissolved lead concentrations from the lead carbonate trials met or exceeded the lead concentration in the unfiltered sample, indicating that practically all of the carbonate had dissolved. However, the dissolved lead in the brass shaving trials averaged only around 57%, indicating that there was a fairly low recovery rate. And so the lead test strip kits we used included a plastic well to hold the water, an eyedropper, and the test strip. We followed the manufacturer instructions to the letter for the unacidified trials, which said to add 7 drops of water to the well, swirl it for 1 minute, then introduce the strip and let it sit for 10 minutes before taking results. For the acidified trials, we first added 6 microliters of 0.5 m citric acid, sodium citrate, to the water in the well, and then we later let the well contents incubate for 40 minutes, as said previously, before adding 14.4 microliters of 1 m sodium bicarbonate to neutralize the acid. And without this step, the acid would interfere with the reading and it would not provide any usable results. So we let the strip sit there for 10 more minutes after the sodium bicarbonate step, and then we photographed the results with a cell phone camera using a standardized method as seen here. So, as mentioned previously, the lines on the test strip will reflect the lead concentration in the water. So the darker the right line compared to the left, the more lead was detected in the sample. So we used the free image processing software ImageJ to convert these images into numerical data. First, the images were uploaded into the software and they were cropped to only show the lines on the test strip. These images were rotated to make the lines as close to pointing directly upwards as possible, and to accentuate the difference in color between the lines, the image was split into 3 grayscale versions representing the red, green, and blue color channels which compose the image. And we determined that the green color channel would be best for enhancing the 2 lines. Next, we used the plot profile tool in the software to gather data on the average color of each column of pixels across the image. And prior to this step, the selection tool was used to further cut down the borders of the image, leaving only the lines. Finally, the data was copied into a spreadsheet where the local minimums on the graph were located. So the value representing the left minimum was divided by the right to make a ratio. And essentially, the lower the gray value on the y-axis, the darker the color of the strip. So here are some of our results. As seen on the left graph, the acidification method did not interfere very heavily with detection of dissolved lead. There was a slightly lower. I'm blanking on the word. Why am I blanking on the word? Okay, sensitivity. As can see, there was a slightly lower y-intercept for the acidified samples, but overall the R-squared values and the slope were relatively unaffected. And on the right, you can see the lead carbonate trials. For the unacidified samples, there was actually a negative slope close to 0 and a very low R-squared value, which indicates that without the acid, it was unable to detect these lead concentrations. But for the acidified trials, you can see there's a much higher R-squared value and a slope that more closely matches the dissolved lead detection. So this indicates that our acidified method was able to improve detection of small particulate lead, or at least lead carbonate. Ooh. What is happening? Okay. Now we can see here the brass trials. Compared to the lead carbonate trials, there was actually not that much of a difference between the unacidified and acidified trials. There was relatively similar and low R-squared values and similar slopes, indicating that our method actually did not improve the detection of large particulate lead. So for our next steps, we're planning to run additional replicates of lead trials to improve the methods, especially to improve detection of brass. As mentioned previously, our results were less than promising on that front. And we want to narrow down a formula which can relate test strip ratio to lead concentration for use in a mobile app. So this app called Wellware is already functional and actually already on the Apple and Android stores as of today, and it's currently in testing. So it can be used to take images of test strip results and perform the ImageJ method to compare the color of the strips to determine whether a homeowner is at low, moderate, or high risk of lead contamination in their water. We hope by refining the method and formula, this app can make it easier to accurately test for lead in their own homes. We're also developing field testing kits so researchers can collect water samples from community members and test the lead content outside the lab. So these testing kits include quarter-liter bottles with citric acid already added and a dropper bottle with sodium bicarbonate formula. So we're in the testing phase with this field testing method, and results so far have been promising. So this image is of a lead test strip which was used with one of these field test kits with water containing lead carbonate. And as can be seen here, the right line is far darker than the left line, and image processing yielded a line color ratio of 1.27, indicating that the strip detected a high lead concentration of lead carbonate, which it's unable to do without the acidification method. And for work on the study, I'd like to thank my professors Michael Fisher and Amanda Northcross, as well as Dr. Zhenfa Zhang, I think that's how you pronounce it, who helped me with running ICP-MS spectrometry on the results, as well as my lab assistants Angelina Klepp and Nancy Smart, and the Office for Undergraduate Research, which assisted with funding.
Thanks so much, Ethan. Questions for Ethan?
I might have missed it, but what is the detection limit?
Oh, yeah. I believe it's about 3.8 micrograms per liter for the test strips.
Okay.
And that, with our deacidification method, because of the lower sensitivity, that might have increased, but we can adjust for that using the slope, as we mentioned.
Well, thank you, Ethan. That was a wonderful job. I was curious about, have you had any false positives of testing water that doesn't contain lead, but maybe other water chemistry parameters like high turbidity or high DIC or other metals that indicate that it may contain lead?
We haven't been able to test for a large variety of water samples. Mainly, we've been alternating between deionized water and then standardized test water using an EPA method. As for false positives, they happen occasionally, but they're fairly rare, and it seems more a problem with just a batch of test strips as opposed to the method. Like sometimes we'll just get a box of test strips where all of them don't provide any results whatsoever, and sometimes they'll just get an entire box that provides false positives. But I mean, this was already covered in previous data that these cheaper at-home methods do have a lower precision.
Great, thank you.
Maybe my follow-up question is about the sensitivity of the ImageJ analysis. Did you do any sensitivity analyses on the code? Could the software accurately distinguish between a positive and a negative, and does that play into your method of detection?
As in for the, okay, I'm trying to, there's a delay on this when I press the button. I don't know what's going on. Anyway, so, yeah, as for sensitivity, we just basically been assuming that the ratio of the left line to the right line would provide that anyways. Of course there is some variation.
Okay, great, helpful. Any other questions? Thanks so much, Ethan, really appreciate it. Next, we'll introduce Kyle Rezek, who is a PhD student at the University of North Carolina, and he will be presenting to us his progress in developing standardized methods to quantify lead composition of drinking water system infrastructure using X-ray fluorescence. Kyle?
Thank you, Nita. Hello, everyone. Thank you all for coming. It's nice to see some familiar faces in the audience, and thank you for all your support. I'll be talking about using an XRF to determine lead composition and verify whether it contains lead or not using X-ray fluorescence. And I'd like to thank my colleagues Timothy, Lily, Elisa, Sid, and Mike. So overall, as you are most likely familiar with, lead causes a variety of health effects, especially neurological effects that impact behavior, growth, and behavioral problems. There has been no known safe level of lead identified, and drinking water can be a main route of exposure, as well as the air, soil, food, or dust. And the WHO, though, has a drinking water guideline of 10 parts per billion, which is a progressive improvement that is used to try to reduce the amount of lead overall in drinking water. A recent systematic review that was done here at the Water Institute looked at the different concentrations of toxic metals in low and middle income countries from different sources of drinking water across the world. And especially in sub-Saharan Africa, it was shown that a lot of the samples observed were above this WHO guideline of 10 parts per billion for lead. And then some other samples from around the globe as well also exceeded that standard. So this is not just an issue based on one water system or one country, but it can be found all over the world. And it's important to have prevention methods, which is one of the goals for motivating this project. The International Plumbing Code, the IPC, and the National Sanitation Foundation have standards that limit the amount of lead that could be in a product of 0.25% or below, and it could be considered lead-free. Recent studies have shown that both tanks and taps that are used in rural water systems that are usually made out of brass and galvanized parts contain lead levels much more above this 0.25% of lead content, and stainless steel has been shown not to have lead in either very negligible or non-detect levels. And so this is what the brass tap and galvanized hand pump spouts usually are in the field, and we are recommending stainless steel taps and hand pumps, which have shown not to contain lead by XRF. And we tested this theory through previous work on lead leaching in the lab using NSF 61 protocol, showing that brass taps had the largest amount of lead leached over a 3-week dump and fill test with very aggressive test water. And on average, they had around 200 parts per billion. The galvanized spouts had around 40 or 35 parts per billion. And the stainless steel spouts, taps, and PVC that was here purchased from America that did not contain lead and was certified by the standards all did either negligible or very low amounts of lead. So overall, the standard right now is destructive testing, where you get shavings of the material and you test that on an ICP-MS to determine if it has lead. Though this is very difficult, though is the main standard that is used. These lead leaching and performance tests that were previously done are also difficult, expensive, and time-intensive. Though they do inform about prioritizing hazards and using them in different water systems, they're not suitable for verifying that they either contain or do not contain lead. So overall, if you were at Timothy's presentation on Monday, you were informed that prevention starts with the supply chain. And a recent study showed that a switch from galvanized to stainless steel components was about 2% of the total installation cost, and from brass to stainless steel was about 1% of the total installation cost. So if you just look at each component individually, it may be 2 or 3 times as much, but over the entire course of the budget of the system, it's only like around 1 or 2%. So overall, we know that lead in drinking water is preventable by not using lead-containing parts, though prevention requires fixing the supply chain, and we can't fix a supply chain without good verification monitoring. So this comes where the XRF comes in, and this is a handheld X-ray fluorescence device which uses X-rays to determine the material elemental composition. It can scan a variety of materials such as alloys, metal, plastics, and soil. It reports the concentration either as a percentage in high amounts or as parts per million, which is 1/10,000th of a percent. Overall, there are some limitations. It can be very expensive. Some units run from like $30,000 to $50,000. It doesn't give exact performance data like leaching tests. It does produce a minimal amount of radiation, though that can be reduced significantly with proper PPE. It is less precise than destructive testing, and there are possible concerns with curved, rough, and non-uniform surfaces. Though some of the strengths and benefits is that it's very quick with instant results from around 20 seconds to a minute. And if you're able to use this for many, many field tests over a several year period, the marginal cost per test will end up being very low. It is sensitive enough for verification monitoring, and best of all, it's portable, sturdy, and can fit in a briefcase and be transported around in the field, and is very durable in a wide range of conditions. So our proposal and idea is that you can use the XRF to be used to test and verify whether this water system components contain lead greater than the 0.25% standard, and if so, you could decide whether or not to use it in the field. And our key question is, can this XRF device be used for reliably determining the presence of lead in these water system components? And we did that 3 different ways by comparing the percent of lead on the inside versus the outside. Since you scan on the outside and the goal is to have a rapid throughput method, if you have to then destructively test each component by seeing on the inside, whether the outside is representative would help making that process much faster. Whether there is homogeneity of the scans over the product surface area, meaning that can you just scan in one location and that's representative of the lead content over the entire surface area, or should you do multiple scans and set intervals throughout the length of the material? And what protocols, methods, or recommendations are needed to maximize the impact of the XRF method? So what we did in my lab was we used the SciAps X200 handheld XRF to scan the materials. There was calibrated before each scan, and each surface was cleaned with alcohol wipes, and each component was scanned in triplicate for determining error bars. And different radiation prevention and was used, such as a lead shield similar to that you would use at a dentist for x-rays. We also had as well, and all operators were behind this shield. And we had different sets of materials shipped from both Ghana, of PVC from Ghana, including taps of brass and stainless steel, and spouts of stainless steel and galvanized, and then 4 different types of plastic pipes from Uganda. For testing the homogeneity of the brass and the galvanized parts, for the brass taps, we tested the body of the tap, as well as both the effluent and influent sides of the fittings. And then for the galvanized spout, we tested the 4 different sides, the front, back, bottom, and top, and then 5 centimeter intervals throughout the length of the spout. For the inside versus outside, our goal was to get a 1 by 1 inch little sample that we could fit into the test station seen on the left, which stabilizes the XRF device, minimizing error and bias of moving the device or the sample around. And a bandsaw and a handsaw were used to cut these samples, and these were scanned in the test station. For our results, it was shown that the brass taps have a wide variation in the lead content, and all 3 taps that were scanned were statistically significant in their difference of lead composition from the influent to the body to the effluent. Overall, like the influent had around 2.6% on average, the body had 3%, and then the effluent, which is where the water comes out of, is around about 4.5%. For the galvanized spouts, only one of the spouts was statistically significant in the lead difference over the surface area, though I want to definitely point out that for the spout 2, if you scanned on the welded line, it gave a value of 0.25%. So this is a great example that if you were only to scan by circumstance at that one point, either intentionally or malintentionally, it could be that you'd say, oh, this piece meets the standard, and you would accept that into your system. Well, if you look at any of the other scans on that second pipe, it shows that there was a range of 0.25 to 0.55% of lead. So you could also be introducing other parts of lead. While the other 2 spouts had a maximum of 2.6 and 2.8 or so percent of lead. For looking at whether the inside versus outside was representative, we graphed the outside percent lead on the x-axis, and the y-axis was the inside lead percent, with the line in the diagonal showing y equals x, meaning that if it's on that line, then it's representative and about similar. And then anything in that top right quadrant was exceeding the 0.25% threshold and not passing that standard. And both the stainless steel spouts and taps were non-detect for lead. So overall, the Uganda HDPE and polyethylene pipes were very much below the 0.25% lead content, while the rest of the components were above the 0.25% lead. Zooming into that quadrant, we see that the PVC from Ghana had about 0.3% of lead. An unknown type of plastic pipe from Uganda was around 0.6. The galvanized spouts were on average around 2%. One model of the brass tap was about 2.5, and another model was 6%. So it definitely shows that if you look at different types of brass taps that are manufactured by different factories, one could have 2.5 and one could have more than 3 times as much of lead. From our preliminary lead leaching tests, we see that the PVC from Ghana over a 3-week test period leached 23 parts per billion of lead. The galvanized spouts, as mentioned previously, leached around 34, and the one model of the brass taps leached just under 200 parts per billion of lead. So why does overall this all matter? Measuring heterogeneity matters because lead leaching tests don't measure heterogeneity and integrates the total lead release over the entire surface area rather than just determining if a certain spot is a hot spot and contains higher lead than other areas. And 1 out of the 3 of the spouts and all 3 of the brass taps had statistically significant in their difference of lead. And it was shown that the scans using the XRF is representative on the outside as it is on the inside, where water is in contact with the material, and that's where we are focused on. And this doesn't necessarily require destructive testing for every component. So some recommendations we have that are preliminary, if you're interested in using this in the field or seeing if your products contain lead, is that we recommend scanning the component in at least 3 different locations and scanning at least 3 different components that are individually to see if the lead content varies over the product line, and scanning all locations in triplicate as well. And if scans are less than 0.25%, then overall it should be okay to use. Though if some scans or their 95% confidence interval from the at least 9 different scans, then we recommend maybe doing additional testing of replicates, locations, or maybe doing lead leaching performance tests if able. And overall, this could help improve the supply chain to prevent lead exposure in drinking water, which overall could lower the blood lead level and improve the health of these communities. So some future work, we're looking at obtaining the shavings of both the inside and outside, running them on ICP-MS and verifying against the lead scans that we got, and testing additional water system components and different parameters such as the duration of the scan angle and whether it's a rough or smooth surface and whether that introduces bias to the x-rays being scanned. Here is the references from the presentation, and I'd like to again thank my colleagues and all those that helped shipping the parts over from Ghana and Uganda. And there's a corresponding poster that some of my colleagues have worked on. And I want to plug the Global Lead-Free Water Pledge, which is where we are trying to promote lead-free infrastructure and better health of different communities. So thank you.
Thanks. Questions for Kyle?
I have a question. Did I understand it correctly in that the concentration of lead on the outside of whatever it was you were measuring, the spout, that the outside reflected what we saw on the inside?
For the most part, it was. There was one galvanized spout, the second one, that it was not on the line at all, while all the other ones were either very close to the line or directly on the line. And that was also the spout that had that 0.25% of the scan on the welded line. And it kind of shows that overall, it may, especially with the galvanized spouts, they're a little more untrustworthy because it uses the zinc dip bath for galvanizing the iron steel. So overall, there could be fluctuations in that zinc bath and then of the lead content. So the galvanized spouts, we recommend maybe doing a little more extra scans. So, but yeah, overall, it was about representative on the inside versus outside. So we could say it might be the same material being used to manufacture it. Thank you.
Well, we're on this slide too.
Yes.
I'm curious, you said PVC had the lowest amount of lead on the inside and outside. where does that 23 parts per billion come from? Is it the water source, or?
Yeah, so from the PVC from Ghana, we did a lead leaching test of using corrosive water. And we think the lead in PVC, especially in Ghana, is from like the thermal destabilizer to make it more kind of hardy out in the field and not degrade as fast from thermal exposure or heat or the sun. But yeah, the 23 parts per billion was from the lead leaching test that we used to expose it to water.
Roger that. Thank you. Good presentation.
And you had a question?
Yeah, thanks. Very interesting. You started by saying that this is a very expensive technology, and I was wondering how expensive it is, because again, if it would work for a long lifetime, maybe the per unit costs aren't so much. So also, if you know how long, what the expected
Sure. So usually they cost between the range of like $30,000 to $50,000, depending on what kind of add-ons you can get, and especially different calibration models. So if you want to add soils, or you want to add plastics, or different tap suballoys, or different elements, it could increase the price. Overall, it may need to be sent back for calibration in the factory every several years, but overall you can use that as long as the battery is charged. The battery lasts 6 to 8 hours, and you can just keep using it and using it. And overall, if you, I mean, you could do the math, but if it takes 30 seconds per scan and do 8 hours of battery life, you can do many, many samples, which comparative to ICP-MS, which might be much more expensive per sample overall. So it would be a large initial investment, but over time, if you're able to use it, it could substantially reduce the cost over time.
It looks really easy too. So.
Yeah, you just point and scan, yeah.
Last questions for Kyle? Okay. You can find him at the poster event as well. Thanks, Kyle.
Thank you, everyone.
And next we'll welcome Sooyeol Kim, who is a postdoctoral scholar at UC Berkeley, and she will be presenting to us today her study examining wastewater for patterns of antimicrobial resistance across the US.
Hi everyone, I also go by Susie, and I'm a postdoc at UC Berkeley in the Pickering lab. And I'm excited to tell you a little bit about our study today, but we only have 12 minutes, so I'll go a little fast, and the plots might be slightly small, but you can also check out our preprint for better resolution. Okay, so I just want to go briefly over how antibiotic resistance is this silent pandemic that doesn't get enough attention as much as the COVID-19, for example. But it affects us globally, and the US reported more than 2.8 million antibiotic-resistant infections annually, resulting in thousands of deaths. And this is actually a statistic from 2019 CDC report, and since then, we've only seen increases in these clinical isolates. So here is a table from their 2022 fact sheet that compares the 2019 numbers to the 2022 numbers for different threats classified as urgent versus serious for various antibiotic bacteria that's seen in clinical settings. And you can see that there's a big increase. And recently, CDC also produced a report that showed that certain antibiotic-resistant bacteria in the last few years have been increasing significantly. So in order to really look at antibiotic resistance and know which targets to prioritize, we need to have good surveillance methods. And currently, the surveillance methods for antibiotic resistance really involve clinical surveillance, which can be limiting because it's biased towards people who are actually seeking healthcare. So that might exclude people who don't have active infections, or don't have the time and resources to actually visit clinics. It's also individual samples, so it takes a lot of resources to get a community metric, and results in delayed monitoring. On the other hand, you have wastewater monitoring, which can be a good complement to this clinical surveillance. And this captures antibiotic resistance profile at the population level. It includes not only the hospitals, but the community around it. And it gives us real-time monitoring, which can be ideal for this invisible pandemic that we're going through. So in our study, we wanted to leverage the existing wastewater surveillance system that we have in the US for viruses and do take a snapshot of the antibiotic resistance genes that's in our wastewater to look at these clinically relevant genes and how they're distributed across the United States. And to my knowledge, or to our knowledge, this is the first study of its scale that gives us quantitative information and cross-sectional concentration data for the US. And then we also wanted to see whether there are any characteristics that are associated with these sewer shed abundance, to see if there are any characteristics that we can use to predict wastewater antibiotic resistance gene concentrations. So to do this, we selected 11 different targets, and here you can see our list that's for various antibiotics, and applied these assays across samples from 1 week, which then we could turn into a total resistance burden metric using z-scores, and then combine that with various secondary data sets that were potential drivers of antibiotic resistance. So for example, antibiotic prescription data, socioeconomic factors, and animal agriculture, and then points of interest like nursing homes and hospitals. And this allowed us to assess the correlation and also construct prediction maps using random forest modeling. Okay, so I'll go briefly over a few of our results. And first, to look at the regional differences, we divided up 168 wastewater treatment plants into 4 different census regions. And here I'm showing you the burden score and the total resistance on your left, and then the beta-lactamase resistance on the right. And you can see that there isn't necessarily a lot of significant differences within regions. But this is also because when you look at individual genes, they differ, the difference in their trends have opposing effects. So when you actually summarize everything and lump everything into one metric, it sort of obscures those patterns that you see. And we were also able to expand our wastewater data into different regions as well, using the secondary data sets that we mentioned through modeling. And again, our antibiotic resistance maps show more insight when you examine individual genes instead of the total resistance metric. And this really reflected the importance of looking at individual genes, but also the difficulty in coming up with a metric that can describe antibiotic resistance as a whole when there are so many different antibiotics and different mechanisms in play. So next, I'm going to show you a series of correlation plots, and the top box is going to show you resistance burden to antibiotic class, and then the bottom box is going to show you correlation with specific antibiotic resistance genes. And here I'm showing you the correlation with antibiotic prescription and different antibiotic class on top. And contrary to what we think we'd see, we actually saw very little correlation with the wastewater genes. And this can be potentially due to a discrepancy between antibiotic prescription data versus what we actually consume for both humans and animals. And in our random forest modeling, we were also able to look at the order of importance of these variables in constructing our models. And out of the 28 variables that we used, you can see that antibiotic prescription data variables ranked pretty low. And related to that, we also looked at number of hospitals and nursing homes. And you can see that there wasn't a significant association with the number of hospitals and nursing homes either. And those variables also ranked pretty low in our random forest modeling as well. Next, I'm going to show you a whole bunch of socioeconomic variables, but I'm going to only point out 2 of them that had some prominent correlation, and that's proportion of people that are under housing burden and also proportion of people who don't have insurance. And these 2 variables were also the top 2 variables that were most important in our random forest modeling as well. And this really reinforces the idea that risk of antibiotic resistance is linked to socioeconomic disparities, which has been shown in literature as well. And lastly, I'm just going to go over some race and ethnicity variables. And you can see here that being of a certain race, you had associations with certain genes in the wastewater. And these variables were also significant in our random forest modeling as well. And we aren't necessarily saying that race is associated with antibiotic resistance, but more that race can be correlated with certain systematic disadvantages like access to healthcare that's been proven to, or that's been shown to have strong links to antibiotic resistance. And then we also looked at limited English proficiency, which we interpreted as having stronger ties to a home country, so either as a sign of frequent travel or recent immigration. And together with the variable of airports, presence of airports in the sewer sheds, we showed, or we agreed with a certain literature in that international travel may contribute to antibiotic resistance and the global spread of these genes. So the final takeaways from this study were, first, wastewater can be used to look at antibiotic resistance genes, and these results here can be used as baseline for assessing temporal trends and other interventions that we may see in the future. Certain social vulnerability factors like housing burden and lack of insurance, and potentially travel patterns were associated with wastewater ARG prevalence stronger than antibiotic prescription rates. And lastly, we contributed to a growing body of literature that showed that antibiotic resistance burden really disproportionately affects low-income and disadvantaged communities. So all in all, I think the study really shows that in order to fight antibiotic resistance, you need holistic approaches and not just antibiotic stewardship that involves raising the standard of living and increasing the access to healthcare in order to really effectively fight this silent pandemic. And as always, there were multiple people that were involved with this study, so we'd love to thank all the co-authors that I showed in the beginning of the slides, and also our funders and Wastewater Scan, which is the National Wastewater Surveillance Program that the US has, and of course our wastewater treatment plants for providing us with the samples. And this is a QR code for our preprint, which has not only plots that you can actually read the labels of, but also more discussion and analysis of our data. It also shows a plot of the regional distribution of all the different genes, which I've sort of heard is of interest. So make sure to check it out, and it's currently under review for publication right now. So hopefully you will also see it as a publication soon as well. Thank you for your time, and I'm happy to take any questions.
Questions for Susie?
Thank you, Susie, for the excellent presentation. You mentioned that you were looking at clinically relevant ARGs. The trick, though, is that you don't know the source of the ARGs in the wastewater. Do they come from maybe biofilm in the sewer, animal sources, and so on? And you looked at a really nice list of socio-demographic variables. Did you consider collection system characteristics? Sort of, I would think, like average residence time or retention time in the collection system itself, maybe whether it's a combined or a separate system, that kind of thing. Did you look at that at all in your analysis?
In this analysis, we didn't. I do think it would be really interesting to look at like combined versus separate sewer systems. But for this, because we were working with Wastewater Scan, we just took kind of all their sample data and applied it here. But I think it's a good idea to definitely look at those as well. Yeah.
Thank you.
I have a question. Thank you for an awesome presentation, Susie. When you mentioned that a big portion of unhoused population was associated with high concentrations, I am wondering if those sewer systems were combined or only like from the pipes, because I'm wondering how did let's say like the samples from the unhoused population make it into the wastewater treatment plant.
Yeah, just to clarify, the housing burden is actually defined as people who pay more than 30% of their income for houses, whether that be rent or mortgage, utilities, and so on. So we wouldn't necessarily be able to say something about like unhoused population, whether they're connected to the sewer system and this analysis in general, but it refers to people who are having trouble sort of in managing their money, I guess, or yeah, and having to spend a whole portion of their income on their housing.
I think I was understood. Thank you.
Other questions for Susie?
Sure, I'll wade into this. So Amy and Susie, really interesting work, thank you. As I venture into this AMR world, it seems to me like it falls into the PCR is good enough versus you have to do culture and then PCR. And I just wondered if you want to discuss the reasons why you decided to only go with PCR and how that could be an advantage or a limitation for your study.
Yeah, I think both PCR and culturing have advantages to its own. PCR is great because we can do it really fast, whereas culturing takes hours to do or days to do. But on the other hand, genotype, so whether bacteria have these genes, that's not necessarily going to actually translate to having a phenotypic antibiotic resistance. So culturing is also important in knowing that yes, these genes are actually being translated into having antibiotic resistance for these bacteria. But I think knowing that these genes are present in the environment is still really important because the bacteria has to have these genes in order for these phenotypic traits to come about. So this is kind of gives us the background for knowing, oh, there are a lot of, for example, NDM in our environment, which we didn't expect to see based on like our clinical data. Then people can really go in and test for bacteria that might have that resistance. So I think you can have sort of a scaffolding system where PCR is an easy, fast way to screen for these genes, and then do culturing to get additional insights into antibiotic resistance.
So if I could just add on to that, what do you see as the next step in this line of work that you're doing?
For additional work on this, we would love to do like longitudinal studies to see how how the different trends in PCR data goes. But we would love to also couple it with culturing data from wastewater to really get that additional insight as well.
I mean, I think we're moving towards culture-independent methods broadly. And so I think this study the idea was to see what can we find out about AMR using digital PCR, which is very quantitative, and we can look at concentrations very accurately. And I think our next step, as Susie mentioned, is to talk with SCAN about longitudinal monitoring and seasonal fluctuations, but also we're working on methods to look at genomic context of ARGs to be able to identify what bacteria are carrying these genes. I think that's kind of like the next step here, using culture-independent methods.
Yeah.
Thanks, Susie. Great, and luckily we get to continue these conversations with our next presentation. So I'll invite up Nita Khanal, who is a PhD candidate at the University of North Carolina in Charlotte, and is going to describe another approach for quantifying AMR genes in wastewater, particularly in rural environments.
Thank you. I'm Nita Khanal. I'm PhD candidate in UNC Charlotte in Infrastructure Environmental System. So today I'm going to present on the topic to quantitative and digital PCR-based detection of antibiotic resistance gene in a small-scale wastewater system. So I can say this may be a little bit extended version of previous presentation. Why it's not? moving from here. Excuse me, it's not moving from here, sorry. Yeah. I couldn't do the slide, so.
No, I have a feeling, and if I can't pull it up, then we'll get the tech to come in so you have your notes with you. A brief intermission.
Do you want me to call the tech?
It might be okay. Let's see if we can try this again.
It's not showing the full time. If I put the cursor here, then it's not moving.
Does that look okay?
I just want to see the full screen here.
Can you move the microphone down too? It's hard to hear back here.
Yes, yes, okay. Okay, let's go with the antibiotic resistance genes. So it's already previously mentioned. As you can see in picture, the antibiotic resistance genes are genetic component that is found inside the microorganism that can resist 1 or 2 antibiotics at a time. And it's the silent pandemic because it is spread globally and killing millions of people, but it is not noticed as the SARS-CoV-2 is noticed by the public and also the healthcare facilities. And this is a global concern because if resistant organism is there, then it can lead to the treatment failure, it can increase the healthcare cost if we need to stay for a hospital for a long time, and it easily spread in communities, and ultimately it can increase the mortality. And wastewater treatment plant is hotspot for the antibiotic resistance gene because it can be accumulate from various sources, right, like agricultural wastewater, municipal wastewater, and it won't go away even after the treatment in the wastewater treatment plant, and it will come to the contact in the human by the various means. That's why this is the hotspot, and it is having higher concentration even after the treatment. And when we talk about the antibiotic resistance gene in wastewater, we mostly focused on wastewater system in the large-scale or urban wastewater treatment plant, and small-scale rural wastewater treatment plants have very limited study, and we can say it's not well documented. And also several research says that it is less influenced by the human activity. That's why it can help identify emerging trends in antibiotic resistance gene and detect the spread of new resistance mechanism in the rural system. The antibiotic resistance gene that we selected for our study is carbapenem resistance and vancomycin, which is the carbapenem fall under the urgent threat by the CDC, and vancomycin it is fall under the serious threat by the CDC according to 2019 data, and which is updated in 2021, I believe. So our study is mainly focused on ARG detection by using both qPCR and ddPCR. Both detect and quantify antibiotic resistance gene in their own way. For the qPCR, it monitors DNA amplification by using the fluorescence. For the ddPCR, it partitions into the thousands of droplets and give the absolute quantification, whereas for the qPCR, we need to make the curve so that it gives the relative quantification. And qPCR, it is generally more prone toward the inhibitor, and ddPCR, it generally resistant to the inhibitor and don't have inhibitor. But the advantage of the qPCR is it has lower cost as compared to the ddPCR and also give the higher throughput. And for the ddPCR, it has higher cost and give low throughput. This is the objective and hypothesis we said. Objective is to conduct the surveillance of selected ARGs in small-scale wastewater treatment plant, and to compare the performance of qPCR and ddPCR for the detection and quantification of ARGs. And hypothesis, we said ddPCR will demonstrate higher sensitivity and precision than qPCR, particularly at low zinc concentration. So this is the workflow. We collected samples from 7 different small-scale wastewater treatment plant from NC. We collected total 70 samples. And we did electromagnetic filtration for the concentration of ARGs from the wastewater. Then we did DNA extraction by using MagMax wastewater, and we use wastewater ultranucleic acid kit. We use gyrosine as the extraction control, and then we perform qPCR and ddPCR to detect and quantify ARGs. So our ARGs target, we had 3 ARG targets: KPC, OXA48, and VAN-A. Also, I want to say that that this work is collaborated with UNC Charlotte and UNC Chapel Hill. So we in the UNC Charlotte we worked on qPCR, and UNC Chapel Hill we worked on ddPCR. So that's how we are working, collaborating together. For optimization and of the qPCR and ddPCR assay, I can say that for qPCR assay optimization, it was a bit tedious than the ddPCR assay, because for the qPCR assay optimization, we first adjusted annealing temperature and also the time, and then we tested 19 different primer probe concentration combination, and if we find that if that is optimized, then it was okay. If it is not, then we again optimize the denaturation time and temperature to get the optimized assay. And for ddPCR assay optimization, we adjusted annealing temperature gradient, and rest we either follow default or we make constant. For the primers and probes, it was default with the ddPCR setting, and also default thermal cycle condition. So this is the standard optimized curve for the qPCR for all of the targets. So efficiency is more than 90% to 110, and the slope is 0.99, which is optimized. For the quality control, we use positive and negative quality control, and for the negative quality control, we use all the negative quality control in filtration, extraction, qPCR, and ddPCR. Also, we ran limit of detection for qPCR and ddPCR both, and we use the positive control for qPCR and ddPCR, and for especially qPCR, we generated the standard curve by serial 10x dilution. This is what we get our result. So if you see the table from total 69 of samples, we detected KPC in both ddPCR and qPCR. And for OXA48, we found only 1 negative for qPCR and all positive for ddPCR. But for VAN-A, if you see it here, we found only 22 positive in ddPCR and all negative in qPCR, which is kind of interesting. And I'll go over later the concentration of the VAN-A that we got. And you can see for the KPC, for ddPCR and qPCR, there is no any significant, so there is significant difference between the ddPCR and qPCR. That means ddPCR is a little bit more sensitive toward detecting the ARG target. And for OXA48, we didn't find any kind of significant difference, though you can see the median is a little bit higher in the ddPCR than the qPCR. We also did the correlation plot between the qPCR and ddPCR. For both KPC and OXA48, we can see very strong correlation in between them, which means the both methods are aligning and directly proportioning to each other. So here for the VAN-A, we can see that if you see the ddPCR concentration, all are below, like median is below 0.4 copies per microliter, which is very low, and ddPCR is detecting this low concentration while qPCR is not detecting anything, which means it indicates very superior sensitivity of ddPCR in detecting low abundance antibiotic resistance in. And also we compared the extraction efficiency of qPCR and ddPCR. So we find there is no any significant difference between ddPCR and qPCR, but if you see the interquartile range, it has ddPCR has bit consistent distribution as compared to qPCR. And because qPCR is prone to inhibition, we also checked the inhibition test by diluting the samples, but we didn't find any inhibition in qPCR as well, which means the quantification cycle is showing a little bit higher CQ value in both of the target. That means there is no inhibition present in the sample. So from this, what we can conclude, KPC concentration was higher in ddPCR, which indicate better sensitivity. For VNA, it was superior in the low copy detection. For the OXA48, there is no any significant difference we found. And GIRE recovery, it was similar. The extraction recovery was similar for both qPCR and ddPCR, and we didn't observe any inhibition. So take away from this presentation is qPCR optimization required testing multiple parameters as compared to ddPCR. It took month and month just to optimize this target, but for ddPCR, it is not taking that long time. But the ddPCR bit costly than the qPCR, that's what we can say. And the ddPCR optimization happens just with the annealing temperature, and which make faster and simpler workflow. And also, both can perform similarly at the higher concentration, but if we are targeting the lower concentration, ddPCR can outperform qPCR. So that's what the takeaway from this presentation. And I would like to acknowledge my team, my advisor, Dr. Maria Munir, Dr. Rachel Noble from UNC IMS, Dineen, Emma, and funding for the NC Collaboratory, and also those who directly, indirectly support our research. Thank you. Thank you. Questions?
I've got a question. You said that these samples were specifically sourced from rural environments.
Yes.
Could you talk a little bit about the population size of these sites?
So as I mentioned, this is like half of the sample done from the 4 of the sampling site is done from the UNC Chapel Hill, and 2 of the sampling, from the UNC Charlotte area. So one, I know it has this 45 household community, just that, and it is within that range. It's not very large. So now we want to mention it is small scale rather than the rural.
Okay.
Yeah, and I don't that much sure of exact household of each wastewater scale, but it is really smaller as compared to the large one.
Okay,
Yeah.
interesting. And then do you have any plans for these samples to do some analyses, maybe comparable to what Susie has done about
The culture?
yes, yeah, or other like environmental factors, or demographic or socioeconomic factors that might differ between sites?
From these samples, we are trying to check the precipitation versus non-precipitation data as well. Like if we find any difference where there is the heavy rainfall and not, still I'm analyzing the data, but I haven't done that yet. Yeah, that's what we are planning further for analysis. Yeah.
Fantastic.
Yeah, and for culture work and other method, I think we are not going from this project, but yeah, we can think for the later project. This is the first project that we are working, so yeah, we can think about that too. Yeah.
Great, thank you. Any other questions? Yeah, go ahead.
Can you explain a little bit about your jar?
Oh, it is the halophile. So we got that from halophile cell, and it exactly reflect to other targets. That's why we just we are taking that as the extraction control. It is not including in our target, but how much recovery we are getting from the extraction process. For that, we took the gyre as the gyrosine as the extraction control.
Okay, so did you have to like spike it in?
Yeah, we had to spike that before the extraction.
Got it.
Yeah.
Last questions? Okay, great. One last one.
more question.
Yes, Amy.
So you tested for inhibition with the qPCR, but not the ddPCR. Are you planning to do that for the ddPCR, or is there a reason why you didn't?
Yeah, we didn't test it with the ddPCR because it has really I mean, it don't, when we do with the ddPCR, it is more sensitive toward the inhibition. So we already found that if there is no any inhibition with the qPCR, so we are assuming that there is no inhibition from the ddPCR as well. So yeah, we didn't do with the ddPCR. Thank you.
Thank you all so much. This concludes this verbal session. We appreciate our speakers, and we'll see you all at the poster session.